Hello, my name is Ed Hallrett, and I serve as the program coordinator for Rhode Island's IDEA Clinical Translational and Research Hub, which we fondly call Advanced CTR. I want to thank you all for joining me today and for this keynote presentation, and it's my distinct pleasure to introduce Dr. Rodolphe Barrangou, who will deliver the keynote. Dr. Barrangou is the Todd R. Klaenhammer Distinguished Scholar in Probiotics Research at North Carolina State University. He was primarily educated in France, in Paris, and Compiègne, and received his PhD in functional genomics from North Carolina State University and an MBA in executive business administration from University of Wisconsin-Madison. Continuing as a pioneer in the study of CRISPR, Dr. Barrangou spent 9 years in research and development in industry at Danisco DuPont, characterizing probiotics. And there he established the functional role of CRISPR-Cas systems as adaptive immune systems in bacteria, primarily providing protection from bacteriophages. Dr. Barrangou currently heads up a large CRISPR lab at North Carolina State University, and he is the editor-in-chief of the CRISPR Journal. Among his honors, and I won't list all of them, in 2016, he received the Warren Alpert Prize, which has some local significance in that the Warren Alpert Foundation is based here in Rhode Island in Providence. In 2018, he was elected to the National Academy of Sciences in Genetics and in Plant, Soil, and Microbial Sciences. He's also a member of the National Academy of Engineering and the National Academy of Inventors. As you will hear, he's the co-founder of several biotech startups. The title of his talk today is Catching Up with CRISPR Craze, Applications and Implications of a Disruptive Technology. I want to remind the participants to enter their questions into the Q&A, and we'll have time at the end to address those. I'm happy to hand over the proceedings to our speaker.
Thank you, Ed, and good afternoon, good morning, everyone, depending on the time that you're in. I'm delighted to be here. I think this has been a thought-provoking meeting, and I'm confident that this theme and the relevance of CRISPR-Cas systems in general and genome editing in particular will be equally thought-provoking, interesting, and inspiring. So I will indeed discuss today, as Ed just mentioned, catching up with the CRISPR craze, which is crazy as a matter of fact, and discuss the applications and the implications of this amazing disruptive technology. And for the sake of new ideas, right, a very important theme for this meeting, we're all in the business of translating science into products that will impact people's lives, whether they're consumers or patients. And there's a number of steps that are required to do so that starts with basic CRISPR science or knowledge of immune systems, turning that into CRISPR technologies that can be applied to do genome editing in products of importance. And perhaps now more so than ever, we appreciate the need for regulators, the public, the media, and the consumers to accept science and technology. And this is also something I will touch upon. So if we start to look at metrics of really trying to measure the CRISPR craze, assess the CRISPR craze, and look at the CRISPR craze, even from data from 2 years ago, this is a very explosive field with the literature really exploding very quickly upon the advent and the development of CRISPR-based genome editing. It's very diversified for a lot of disciplines, a lot of labs, a lot of industries, and it's very global. If you look at the top 10 countries in the top right that are leaders in CRISPR publications and CRISPR citations, it's noteworthy that it encompasses not just the US, but 5 different European countries, as well as China, Japan, and even Australia, representing Oceania. This CRISPR collective is constituted by 47,000 authors from 4,500 affiliations coming from over 100 different countries that have published 20,000 papers. And if we look at the dissemination, what I call the democratization of CRISPR, since we now live in a post-CRISPR world with genome-edited people walking around to some extent, I'll address that in a few minutes, the numbers are mesmerizing, right? The non-for-profit Adgene, not too far from where you guys are, some of you guys are, has already shipped over 200,000 plasmids to labs, right? Those plasmids have been deposited by over 500 labs, have contributed over 10,000 plasmids, again, shipped to over 100 countries. So this is a truly democratized technology that is changing the world in real time. And if you try to really decipher what's happening right now, it's complicated. It's very diverse, it's very intertwined, it's very interwoven. The good news is that more than 90% of all those papers focus on human cells and human tissue. 90% of those 47,000 authors published on human applications of CRISPR, and another 5% work on mice. So that's great for human health. It's great for the Warren Alport Foundation and people who care about curing disease and whatnot. It also means that only 5% of all those papers do everything else. So I think when we think about ideas, challenges, and opportunities, this is something to take into account as we get CRISPR to the clinic on one side. How far and how fast can we take CRISPR elsewhere? So new ideas, new discoveries. Let's tell the story of CRISPR in that context. And let's start with the science. So this is what an actual CRISPR looks like. And ironically enough, there is no actual CRISPR in CRISPR technology, clustered regularly interspatial palindromic repeats. I'll help you a little bit here to see this. Those are clustered DNA repeats that are regularly interspaced in the genomes of about half of bacteria in archaea around the world that constitute an immune system which captures a piece of an invader, like a virus, like a phage, as Ed mentioned, or a plasmid or transposon or integrase and things like this. And it captures a genetic marker of this invasive element, integrates it into the CRISPR as part of vaccination process, adaptation process, which is genetically inheritable. So bacteria and progeny have the ability to inherit this immune system that's adaptive. And that DNA encoded CRISPR immunity is transcribed into RNA, central to my biology. And those are RNA guides. And those guides literally direct Cas effectors for sequence-specific targeting and cleavage of viruses like phages, or plasmid, or sometimes even self, in ongoing warfare between bacteria and their predators. I'm not going to give a lecture on the amazing, beautiful genetics and diversity of CRISPR-Cas systems. All you need to know is they're very diverse. They're very interesting from a molecular standpoint. There's two main classes, whether the effectors are single proteins or complexes. There's 6 major types, now almost 50 different subtypes. And those effectors sometimes target DNA, sometimes target RNA, sometimes target both. And then the kinds of cleavage and nuclease and or nicking activity that they have is likewise diverse. Think of a different array of molecular scalpels and chainsaws and axes and the like that are readily available for those bacteria to shred, target, cut, and cleave and nick invasive genetic elements. I think that's all we need to know about CRISPR science, amazing immune systems in bacteria that provide adaptive inheritable immunity. The reason I was invited, presumably, and the reason we're here today is because of the advent and the repurposing of CRISPR-Cas immune systems and their effectors for genome editing. This is what was recognized just a few months ago, which seems like an eternity ago in CRISPR years, by the Chemistry 2020 Nobel for Jennifer Doudna and her colleague Emmanuel Charpentier. And this technology, this revolutionary, disruptive technology, has democratized, almost trivialized our ability to target DNA precisely and change and alter the DNA sequence at the site of cleavage using those CRISPR-guided effector nucleases. And the reason why this technology has been so impactful, so revolutionary, so disruptive and innovative and democratized is because it has many advantages. As you can see here, programmable, specific, transferable, efficient, precise, affordable, quick, multiplexable, and scalable, and more. And it's so good and affordable that the average student can use it at the average institution almost anywhere in the world. Now, it's only 9 years old. So like the average 9-year-old is still a little fat, we want skinnier ones. It needs to mature a little bit, throw a few less tantrums, be more reliable, be a little bit more lean, be a little bit more efficient. But the bottom line is 9 years in, it's one of the best technologies we've seen in molecular biology. And 9 years in is good enough already for the FDA to condone human clinical trials in the US. Now, of course, we're going to keep making CRISPR better. There's a lot of opportunities to enhance guide composition, use multiple CRISPR-Cas systems at the same time that are orthogonal, still continue to mine biodiversity and look at PAM targeting and flexibility and precision. That's great. And a lot of people are working on this, and that's fantastic. I think when we think about new discoveries that are pending in the CRISPR space, I think understanding, controlling, and manipulating and predicting DNA repair is one of those things that needs more work on. And then again, when it comes to therapeutics, delivery may be the top 3 challenges that we have to face. And I hope some people in the audience really will help enable and develop novel technologies that will allow and enable us to deploy those molecular machines across the tree of life in general and in human therapeutics in particular. Now, I'm not going to go and do a thorough review of the whole CRISPR toolbox, but in the last 9 years, that toolbox has been expanded and sharpened to a tremendous extent, to the point where now we have the ability to virtually manipulate not just the genome, but also the transcriptome and the epigenome, right? And to do so with multiple different effectors like Cas9 or deactivated versions thereof, or nickase versions thereof, and then Cas3, Cas12, Cas13, Cas transposons, base editing, prime editing, and on and on and on and on. We now have on hand a whole CRISPR toolbox that enables the average geneticist to rewrite the genome, control the transcriptome, and reset the epigenome of virtually any organism on planet Earth. And I would argue that there's no technical limit to what can or could be done, but rather that the only limit is our imagination. And those 47,000 authors have worked very hard in the last 9 years to publish those 20,000 papers to demonstrate the usefulness and the potential of those CRISPR technologies across the tree of life. In academia, at the bottom of the pyramid, people work on model organisms to understand viruses, obviously bacteria and archaea, model systems, model organisms, and that's fantastic. In the real world, where people deploy those CRISPR-Cas technologies and tools for profit and for commercialization and product development, in the food and ag world, we can use CRISPR to make, CRISPR crops, CRISPR chicken, right? CRISPR livestock, CRISPR seafood, CRISPR fruits and vegetables, white button mushroom. And of course, at the very top of the pyramid, again, Warren Alpert Foundation inspired medical applications. I presume most of you are very focused on this, and there's almost no limit to how we can use CRISPR to decipher, understand, characterize disease, and also develop therapeutics that will enable us as a community to fend off disease across the board. So now that we understand that technology to some extent, we have the ability to manipulate, rewrite, edit, control the genome, the transcriptome, and even the epigenome. What are we doing with this? And when I mean we, the collective CRISPR community, right? There's a whole research industry that now caters to the users with great tools and guides and enzymes and softwares and plasmids and on and on and on. You can even order your mouse CRISPR knockout. What merely 6 years ago took a whole PhD to do can now take maybe 6 weeks. At a portion of the cost, portion of the time, higher efficiency and reliability. So it's moved the needle. Now we can use it to advance industry like biotechnologies, any bacterium, any yeast, any algae used in food, biomanufacturing, household care, enzymes, bioenergy, and the like has had, always having, is genome controlled and enhanced using CRISPR technologies. And it's the same in ag already. Plants, crops, animals, livestock, microbes, trees, even flowers and grass, ornamentals, aquaculture, hemp, on and on and on and on. But of course, at the very top of that pyramid is medicine, therapeutics, gene therapies, antivirals, controlling and detecting infectious disease, microbiomes, I'll talk about that, antimicrobial cell therapies, immunotherapies, xenotransplantations, gene drives, diagnostics, big time right now, and pet care, and more. This is a non-exhaustive list. New ideas, new discoveries, all day, every day with CRISPR. And as a matter of fact, if you look at the landscape of CRISPR startup companies, those are not CRISPR companies that are using CRISPR. Those are companies that were incepted based on novel CRISPR technologies. At the very top, some publicly traded companies: Caribou, Intellia, Editas, Beam, now the NASDAQ. On the right, therapeutics: Mammoth, Sherlock, eGenesis, Casper, Excision, Tessera, on and on and on, all the way to Life Edit, Edigene, and Prime Medicine that came out of stealth last week with $300 million Series A and B combined. At the bottom on the right, microbiome control companies: Eligo, Locus, Sniper, and Cilia, and Xbiotics. At the bottom left, ag world, INBRE, pairwise, Trico, and on the left, tool technologies, Caszyme, Arbor, Castag, 12, metagenomic, CRISPR Biotech, and on and on and on and on. Almost, every couple of months, a new CRISPR startup comes up. And it is the democratization of a technology now outside of academia and outside of labs in the real place, in the real marketplace, all the way to Wall Street. And the reason it's so exciting is that it's moving extremely fast. What was like a crazy idea or a crazy dream only a few years ago is now a real medical, clinically tested reality. Go talk to Victoria Gray. All major players in the therapeutic industry have a CRISPR strategy and a CRISPR program with very powerful, compelling, not just science, but narratives talking about patients and saving lives and curing disease. Right? There's 4 clinical trials and INDs right now being managed at clinicaltrials.org. 24, half of them already recruiting actively. Right? Just this past month, the New England Journal of Medicine published exciting results from Intellia on ATTR, in vivo editing of liver cells up to 96% reduction in toxin protein genesis, supratherapeutic level of efficacy, safety and efficacy. And then, of course, as of late, even in my own CRISPR journal, so to speak, precision editing of sickle cell mutations. This is just the beginning of a new era of CRISPR therapeutics and medicines. But this extends beyond everything that we see here. As a matter of fact, the World Economic Forum just recently published a report saying innovation with a purpose, the role of tech innovation in accelerating food systems transformations. When we talk about human health, right? Maybe in the next 10 years, 10 to 100 million patients could benefit from CRISPR-based therapies and diagnostics. But if we deploy CRISPR in crops and livestock, potentially in the next 10 years, we could address 10 times, maybe 20 times as many consumers and impact their health. Maybe it's not 10 to 100 million, maybe it's 1 or more billion people. At a time where inclusivity, sustainability, efficiency, and nutrition and health are all intertwined and critical. Very important. CRISPR is a breeding game changer. It's not me saying it, it's the World Economic Forum saying it, that in the next few years, if you look on the right here, next few years, up to 100 million farms may be planting CRISPR seeds by 2030, generating $100 billion of extra value, increasing yields by 400 million tons, reducing food loss by 20%, and addressing nutrition deficiency. This is why all those people are working not just on humans and mice and animal models to manage health and disease, but also food. Leaner pigs. Ed mentioned, yogurt and phage resistance for more sustainable food agriculture, dairy culture. This is where the CRISPR story started, arguably. White button mushrooms, infectious disease in cattle, and of course, traits in crops. This is a reality. As a matter of fact, 2 weeks ago, the front cover of the New York Times Magazine was a CRISPR-edited tomato. It was from England, I should say, tomato. But very, very compelling, very relevant, very timely, and now in the zeitgeist. Not just in the scientific community, not just in Wall Street, but in our own place. And if we think big, let's just go with trees. So I'm very involved in CRISPR genome editing in trees to breed a healthier forest, but the timelines there are different. When we think about timelines for clinicals, well, it's a 16-year timeline to scale up pine and poplar. 16-year, what we plant today in 2021 will not come out until 2037, right? The scale at which this happens, you can see, is mind-boggling. And the number of people working on this is mind-boggling for the wrong reasons at a time where sustainability and the environment and global warming is a major concern for a lot of us. But let's look at things on the other side of the science spectrum. Actually, in my lab, we work primarily on bacteria, because they're easier, cheaper, and faster, and more straightforward from a genetic and manipulation standpoint. And we work at the intersection of microbiomes, viromes, and CRISPR. And what we do is we realize that the microbiome is big, CRISPR is big, and the virome is not so documented. And we try to develop new technologies that enable us to manipulate them. So at the very top, we do the classical, bottoms-up genetics. what's in the genome? Do you have a CRISPR-Cas systems? Can we find effectors? Can we characterize them? Can we technologize them and then use them for genotyping, phage resistance, transcriptional control, genome editing, epigenetics, imaging and detection, targeted killing for targeted antimicrobials, or even screening? And we've been using those different modalities of CRISPR-Cas systems in farm pathogens, in industrial workhorses, in starter cultures, in dairy cultures, and then in probiotics of importance to human health. As a matter of fact, we're already celebrating the 10-year anniversary of commercializing CRISPR-enhanced starter cultures used to make cheese and yogurt across the globe that have been screened for natural CRISPR vaccination events that make them resistant to phages. And as a matter of fact, that genetic footprint of acquiring a genetic marker against a phage can be used to genetically tag industrial cultures that enable us to tell from a fecal sample maybe what brand of yogurt or what brand of cheese or what bacterium was used to make your food. That can be done. We're also using those technologies to manipulate the genomes of bacteria, and we can be very precise, like we are in human therapeutics, one single SNP, very predictable, one base edit. Or we can use those molecular scissors to take out whole chunks. In the example I'm showing here, we can take out 100 kb of non-essential DNA at a time in a bacterium that's only 2 megs. Think about it, 5% of the genome can be excised in a single DNA manipulation. And then, of course, we've been pioneered and now under clinical testing, CRISPR-specific targeted antimicrobials that enable us to target responsible bacteria agents responsible for infectious disease, like an E. coli or a C. diff or other things like this, where you can use the exquisite programmability of CRISPR to selectively target a genotype of interest in the complex microbiome and eradicate them and remove them. And then, of course, we can use the ability of CRISPR-Cas systems to do genome editing to unleash research, to unleash technology enabling us to understand how bacteria function, who they are, what they do. We can use those molecular effectors to do very simple deletions, stop codon insertions at scale of very specific SNP genesis, remove phages, remove viruses, add markers that enable us to detect them and see them in fecal or food matter in patients or in food services. We now have the ability to use CRISPR-Cas systems across the tree of life to manipulate, control, decipher the composition and the function of microbiomes. And we can do that for women's health, to understand the vaginal microbiome. We can do that in intestinal health, to understand digestion, or health, or inflammation, or colonization, or resistance, or infectious disease, on and on and on. We can use CRISPR typing to understand whether a bacterium comes from the vaginal cavity or the gut cavity, whether it's a poultry species that's chicken or not, turkey or not. And we can also understand when bacteria are transferred hundreds of times in a row or 1,000 generations in a row, how do they adapt to their environment? And how can we reconstitute, change, alter, or engineer evolutionary traits of interest to us to understand what they do? For example, right now we have a big project on controlling and manipulating bile salt hydrolases in the gut microbiome to make it less hospitable for Clostridium difficile. Can we control the enzymatic activity of bile salt hydrolases to promote the colonization ability and the resiliency of beneficial bacteria, and then to really prevent the colonization, the resistance, the persistent, or even the sporulation of Clostridium difficile? Now, I know 2 days ago there was a talk, a fantastic talk, on using CRISPR, not using vaccines to address the current pandemic. And now you can think that we could use some of those approaches, not mRNA-driven, but bacterium-driven, to edit their genome to express COVID antigens on the cell surface of bacteria that we consume orally. People in the US, we already have vaccines. We wish more people use them, right? But if you're in a part of the world where you don't have a -80 cold chain, you don't have access to vaccines, you don't have access to hospitals, the opportunity to have probiotic beneficial bacteria that have been engineered to express those immunogenic markers that can be made in metric ton scales and shipped at room temperature as stable powders is very intriguing. Or we can manipulate those bacteria likewise to control the interplay between the bacterium and the host in the GI tract, the vaginal tract, or the skin to control inflammation, to understand disease, right? To improve the ability of the microbiome to be healthy and to reduce the onset of infectious disease. And as a matter of fact, my lab and other labs are using all those technologies across the globe to manipulate microbiomes in the ocean, in plants and soil, in food cultures, obviously, in livestock and animals, even pets, and of course in humans. Gingivitis, oral cavity, gut health, digestibility, inflammation, vaginal health, infections, BV, skin health, acne. We can use and deploy those various CRISPR technologies to target organisms of interest, to either eradicate them and change the composition, or edit them to change the function and promote health and fight disease. And as a matter of fact, I would argue not enough people are working on this. At the intersection of microbiomes, viromes, and CRISPR, we are already circa August 17th, 2021, at a time where people have used CRISPR in microbiomes to kill phages, people have used CRISPR in microbiomes to edit viruses, people have used CRISPR in viruses to delete or remove bacteria, or people have used CRISPR in viruses to deliver edits to do in situ editing of a microbiome. And as a matter of fact, right now, my lab hasn't published that yet, but we've used CRISPR in bacteria to edit phages. That phage edit is a CRISPR system that is used to control a different host in the microbiome context. So you can see right here, right now, new ideas, new discoveries fueled by CRISPR science and technologies at a time where this is becoming a reality. Whether you're in research, whether you're in business, whether you're a patient, whether you're a consumer, whether you care about medicine or forestry or food or ag or disease, right? There is a CRISPR technology that can be used and exploited to solve a problem that you have. But the tragedy is that there's also issues and concerns. How ethical is it to edit humans? How ethical is it to edit unborn people? Are we going too fast? Are we going too slow? Whose opinion matters or not? How inclusive are we being? Ethical conundrum, the ethics of human genome editing. Of course, PR, public relations, right? Genetically modified people. Skepticism against science and technology at a time where anti-vaxxers are surprisingly vocal and tolerated. Public engagement and debates. Whose voices matter? Are we responsible or not? Are we trustworthy or not? Are we relatable or not? Whose values matter the most? Who's trustworthy in the CRISPR enterprise, in the CRISPR community? And then, of course, regulations. How are we going to regulate CRISPR medicines, CRISPR therapeutics, CRISPR diagnostics? The secure rule last year from USDA, 29 years overdue to update its framework for molecular biology at a time when genome editing is democratized across the world, right? How are we going to manage that? How are we going to promote acceptance of science and technology by the public when it's in their food and or their crops and or livestock? What are the risks? What are the benefits? At a time when people still don't understand basic science, let alone genetics. So there's a lot of efforts underway that I hope we'll get to discuss here in a few minutes to address that. I know I'd mentioned I'm the editor-in-chief of the CRISPR journal, a 3-year-old journal here. And what's mesmerizing to me is that we don't talk enough about science communication as a scientific community. This is what surprised me with the journal, is the diversity of the topics that we address, the diversity and the inclusiveness of the opinions that we relay. In today's issue, it just came out this morning, just before my talk here, right? There's a CRISPR story on people using CRISPR in Pakistan. I mean, think about this, like the trials and tribulations of CRISPR in Pakistan today, right? We all know what's happening next door in Afghanistan, right? I don't know about the CRISPR program, but there's a lot of opportunities, a lot of challenges, lots of issues, right? The last themed issue was CRISPR in China, not just scientifically, but geopolitically. And we talk about gene drives, and we talk about tools, and we talk about ethics, and we talk about science communication, the CRISPR movie, Human Nature, right? There's a lot of applications, and there's likewise a lot of implications. And this is why I hope that no matter where you stand in the audience, on that spectrum and on that level of expertise, whether you're a fundamental academic trying to understand and decipher novel CRISPR-Cas immune systems in bacteria, like I am, that's fantastic. Or whether you're trying to use that scientific knowledge of novel CRISPR-Cas effectors in bacteria or archaea to develop novel technologies and novel effectors to manipulate the genome, the epigenome, or the transcriptome, that's fantastic. Or whether you want to apply some of those technologies to develop medicine, therapies, diagnostics. Or if you're not interested just in human health, you want to do food or ag or biotech, please do so. You may even be bold enough to have a crazy idea, to be entrepreneurial and create a new CRISPR startup company to commercialize those technologies and those products to address the grand challenges of patients and or consumers across the globe. And if you can do all this, or if we collectively can do all this, I will remind you that we still need to work hard on convincing our fellow non-scientists to embrace, accept, understand, or think, or at least talk about the advent and the benefits of CRISPR-based technologies for humankind across the globe. And engaging the public, engaging consumers, engaging the media is more important than ever. So I will stop now to leave a few minutes for what I hope will be an interesting conversation. I think in the spirit of the meeting, I hope the CRISPR craze, the applications and the implication can provoke some thoughts and new ideas, maybe even, dare I hope, new discoveries of or technologies. I will acknowledge on the bottom left the corporations and federal and state funding supporting my lab. And we're very collaborative, so if you're intrigued by what you heard, please reach out to me. I'm also an advisor to a number of organizations in the middle, including the CRISPR Journal and the IGI, and then I have a number of shareholdings and or consultancies with various companies interested in deploying or assessing or commercializing a lot of those technologies. So I will thank you for the invitation. I will thank you for your consideration, and I will preemptively thank you for your pending participation in the Q&A. I'm glad to be here, and Ed, I will let you manage the Q&A session. Thank you very much.
All right, we have a first question. One of the microplastics talks underscored how science communication has to deal with a very different decentralized authority landscape now in the social media environment. How do we surmount this challenge with CRISPR technology?
I mean, that's an outstanding question and a very difficult conundrum. So I think we fail in our education system and training system to prepare people to adapt to today's communication tools. I never took a how to tweet class when I did my PhD. And even when I work at a corporation, I never took a how to communicate on social media orientation class or debriefing class. So we now live in a world where non-experts have an opinion that can disseminate as fast as the CRISPR craze, even maybe faster in some cases. So learning how to develop messages and how to harness those technologies to inform people and make our point is critical. And I think there's two things we need to do. The first thing is we need to become trustworthy and relatable. So we need those Twitterati and we need those social media activists, right? And people willing to do that out there to be likable and relatable. We're not teaching, we're not giving seminars. We have to provide opinions that are science informed, defensible, and also debatable and interesting enough. So we have to market science by using marketing strategies. I think the second thing we need to do is we need to talk more about the why we do it and what the benefits are, rather than the what we're doing and how we're doing it, right? And I think oftentimes scientists, when they talk to the public, they think they're teaching, they think they're giving a seminar, they think they're giving a talk, and they talk a lot about the details that only their fellow scientists care about. And when you talk to a general audience, you really have to adapt your message to the audience to engage them and to relate to them why it's so important to us. Why are we trying to cure disease? Why are we trying to provide a more sustainable and safe planet? Why are we trying to make food more accessible and affordable? I think the how we're doing it kind of is our problem, so to speak, but I think simplifying the message, being more likable and relatable will go a long ways into engaging in the right warfare against people who don't really have the right opinions, just have a loud voice using the wrong microphone, so to speak.
A second question regarding engineering microbiomes. The difference there between engineering a microbiome versus a human is the microbiome is a diverse community. And there's some question about whether in such a community, what's more important, the individual or the function? How do you know that modifying individual species is going to be useful or viable?
Yeah, another great question. So I would, with the preface that as a species, we're also diverse. I mean, at a time of inclusivity here, I want to make sure the audience is reminded we don't all have the same genotype, right? So we are also, even as our own species, quite diverse, though unquestionably we're not as diverse as the microbiome. So the challenge in the microbiome is dual. It's composition and function. Those two things are important. So in the case of infectious disease, if we try to take out the bad guys, right, the select small number of bacteria responsible for infectious disease, I think the intent there is to just remove them, target them, and kill them. CRISPR-based antimicrobials that are specific and efficient really is the key. Changing their function is not the end game. Targeting that phylogenetic unit is the end game. So in this case, it's really the whom or which organism or which genotype. If we flip the equation and we try to enhance function to benefit digestion or inflammation or whatever feature you're interested in, I think there we start to decipher. In some cases, we've made some progress. In other cases, not as much. That's why I talk about bile salt hydrolases as a tangible example that's really legitimate. We can now understand which subset of that population we can manipulate that carries a gene of interest with a function of interest that will yield a phenotype of interest to a patient or a consumer. So for example, as I said, manipulating bile salt hydrolases to control deconjugation of complex bile acids so we can make that environment a more diverse microbiome and less prone to sporulation and function and residence and longevity of Clostridium difficile. So that's where we kind of, know where some of those BSHs are in bacteria like Lactobacillus, for example, and then manipulate them. But of course, this happens to be applicable to bacteria like Faecalibacteria brusnitzii or Bacteroides thetaiota omicron or bacilli or other organisms of interest to the audience here. Clostridium, I think, is underappreciated as far as I'm concerned. So there's a few known, documented, culturable, somewhat editable, and edible in some cases, phylogenetic units in the microbiomes that we know we can target and edit to benefit us, but it's still relatively early in our ability to manipulate the microbiome to enhance and provide and promote health or to fend off disease. It's still early, but we're getting there.
Well, if there are no more questions, I will turn it back over to Ed to finish the session.
And with 2 seconds left, we're on target for the spirit of CRISPR.
Terrific job, Rodolphe. I want to thank you again for a fantastic and inspirational talk, and on a topic that many believe will be the most impactful technology of our time. And it's really inspiring to hear of all of the democratization of this technology, and truly making it available to anyone who has a novel idea to pursue that. And I want to thank the participants today for their questions and their attention, and I hope that you all enjoy the remainder of the NERIC meeting today. Thank you very much.